Okay, we're going to go ahead and start. We're not having a test today. The online test that's set up in there is, I pushed that back because I realized today that I didn't finish everything yet. So I pushed it back for a week. So it's not due. The lab test isn't due until next week. So more fun. And I know we're not going to get anywhere near the lecture test yet. I will never give you a lecture test and a lab test, do it at the same time. So don't worry about that. We're probably 2 weeks away from a lecture test. Like I said, I pushed the lab test back. Now, if anybody wants to take the lab test in class, I can arrange that. Or rather, I should say, if you want a hard copy and not do it online, I know people have had problems with the lockdown browser. In the past, the lockdown browser has caused a lot of grief because it doesn't work a lot. And I can arrange for you to take the test, the quiz, in the tutor center down the hall with Jennifer if you'd like. So that way you can still do an on-ground test, a hard copy in front of you, and not have to worry about, is your computer going to work? Is the lockdown browser going to work? That kind of stuff. Just let me know, and I'll be happy to set you up, and you can take it at your convenience with in the tutor center. It's totally, it's no big deal. It's not a problem. It's up to you. So it will be a different test than the online test, but it'll still be the same content. So, thank you. Okay. So today, then, what I want to do is I finally learned my lesson that you can't do 3 weeks worth of lab in 2 weeks. And I know. It just. I've tried for 4 or 5 years to think I can get 3 weeks worth of lab into 2 weeks, and it just doesn't work. So what I'm going to do is today we're going to do the 3rd week of the Unit 1 lab. We're going to go over the cell cycle and mitosis. We're going to do the cheek swabs. And if we get done in any reasonable amount of time, I may get out a skull and walk you through the first, overview of the bones of the skull. That won't take that long. I mean, I can bore you to tears. I mean, I can show you these bones initially. So in the meantime, let's go ahead and start here. This will you'll hear a more detailed version of this in lecture, but it's always good to start it. Okay, so the cell, we're all made up of cells. We're made of billions of cells. It is the structural unit of all living things. There is nothing smaller than a cell that's alive. You may have heard in the news very recently in Israel, supposedly they created artificial life from fusing two artificial cells together. I don't know anything about it other than what I saw briefly in the news. I don't know if it's growing or not, but supposedly it is some sort of artificial cell. It's not supposed to work that way. Life is only supposed to come from life. Living cells only come from living cells. So anyway, we shall see what they're up to. In our bodies, we have a wide variety of cells. You've seen some of them already when we go through the tissues. We have cells that are flat, and that are irregular, that are round, that can change their shape. They do different things. Some carry oxygen, some carry nutrients, some make up our outer covering. Some carry nerve signals for us. Some contract. The smallest cells are, the very tiny red cells and white cells to cells that are well over a meter in length. The nerve cells that make up the largest nerve in our body, the sciatic nerve that starts here at our hip and runs down behind our leg, behind our knee, and ends up behind our great toe, is made up of tens of thousands of nerve cells. Each an individual cell, each one well over a meter or maybe sometimes even 4 feet in length. That's how long your leg is. They're all different. Every cell is different. One of the things, though, about every cell is that the nucleus of each cell all contain DNA, and the DNA is identical. It doesn't matter what cell it is, the DNA in the cell is exactly the same. The only difference is that the segments of DNA that control that cell are turned on and the others are turned off. The only cells in our body that don't have a nucleus, let me correct myself a little bit here. Red blood cells do not have a nucleus. Red blood cells kick their nucleus out shortly before their development is finished so they can carry more oxygen. That's the only difference. But they had a DNA, they had a nucleus at one point with the DNA. It's okay, You ought to know by now that stuff doesn't bother me, okay? So anyway, so every cell is going to have a nucleus. Not every cell reproduces. Muscle cells don't reproduce. Nerve cells don't reproduce. Red blood cells don't reproduce. Some cells are going to last a very long time. Some cells last a very short time. Muscle cells stay with us our whole lives. They don't reproduce and they have a very long lifespan. The same with nerve cells. We don't grow, as far as we know, new nerve cells. And most of those will last a lifetime for us. Other cells last a few days. Certain white cells that fight infection will last a few days. Some only last a few hours. Skin cells last a few days and then they wear out. They die and then they do the rest of their job of forming a barrier that scrapes away. So again, how long do they last? It depends on what their function is. The cells, even when they're dead, will continue to help us. The basic structures of a cell, most cells all pretty much look alike, at least as far as their structures go. They're all going to have what's known as a cell membrane, the outer covering of the cell. When we look at our cheek slide cells, you're going to be able to see you're not going to be able to see the cell membrane in detail, but you'll be able to see where it is. It's the outer edge of the cell. It's designed to be very restrictive in what goes into and out of the cell. Every cell has a cell membrane. And in fact, every cell, whether you're a plant or an animal cell, or a human cell is going to have a cell membrane. Plant cells have a cell wall around that membrane because plant cells have this problem with losing water. We don't worry about that. We keep our cells either mostly inside our body. Every cell has cytoplasm. The cytoplasm is the goop, the cell sap, the cytosol that the organelles float in. The organelles are these little structures that you see here in different colors here that go inside the cell. The cytoplasm is like a gelatinous mixture, sort of like squishy jello that contains nutrients, it contains proteins and amino acids that the cell needs. And then in the middle of the cell, somewhere in the middle, is the nucleus. The nucleus is an organelle. Yeah, as I said, organelles are like little organs that function inside the cell to do things. Some organelles release energy. Some organelles release antibodies to fight infections. Some organelles produce the DNA, produce proteins. The nucleus is the largest of the organelles. It contains the DNA, which controls the function of the cell. Every cell's DNA is identical. But as I said, depending on where you're located as a cell, what segment of that DNA is turned on. You're going to have all the information for the whole body in your nucleus, in your DNA. You're going to have everything about making brain cells or generating energy or making red cells or telling bone to grow, or repairing, repairing brain cells, for example, and telling muscle cells to enlarge or to shrink. All that's going to be there. Hair color, eye color, skin color, all of your genetic traits, your blood typing. Your earlobes dangle, or are they attached? Something as ridiculous as that. The ability to curl your tongue. That's, by the way, that's a genetic trait. If you can curl your tongue, that's a dominant trait. If you can't, it's a recessive trait. So, and yet that's controlled genetically. Our hair color is a genetic trait. Our body stature is likely a genetic trait too. All this is controlled by the DNA. Now, if you are a skin cell, you're not too worried about hair color, eye color, whether your hair is curly or straight, whether or not that you're going to be producing enough hemoglobin or something like that for your red cells. All you're doing as a skin cell is, I'm a skin cell and I'm doing a good job as a skin cell, and I'm going to last for about 2 days. And then more skin cells are going to push up from underneath. And your role is to protect the dermis from ultraviolet radiation, and to protect the dermis from penetrating injury, and to keep pathogens out. That's all your job is. But your DNA contains everything. You could easily take a living skin cell and transplant it into the small intestine, and it would, if it lived long enough, it would gradually transform itself into a simple columnar cell and start doing absorbing materials. But it has such a short lifespan. Why would you do it? The point is that everything we need to function is in every cell that has a nucleus. And I can qualify it that way. It's just what's turned on and what's turned off. Okay, the nucleus is our control center. The nucleus contains our DNA. The DNA is what we call a nucleotide. It is our genetic makeup. It is a double-stranded molecule, a very large molecule. It's about 6 feet long. Each strand of DNA is about 6 feet long. It is held together with hydrogen bonds. We haven't talked about this yet in lecture, but we're going to get there. The DNA is usually in its unwound state, but when the cell is getting ready to replicate, the DNA will condense into what we call chromosomes, a term we're all familiar with. And we all have 23 pairs of chromosomes. Half of those chromosomes came from our mother, and the other half came from our father. Our cells are busy running through a cell cycle. They have a pattern that they go through. If they're going to reproduce, if they're going to undergo what we call mitosis, then they're going to follow a specific pattern. Mitosis is what is the actual event of dividing the nuclear material, the DNA. You know, we often say that mitosis is cell division. And you know, most people would assume when you say mitosis, you're talking about cell division. That's sort of like the conventional thinking. The correct terminology is mitosis followed by cell division, because mitosis is really nothing more than the replication, the copying of the DNA, so that we end up with a perfect copy of the DNA after the cells have divided. We want to have, you know, the DNA from the parent cell be copied into the daughter cells. And that's what mitosis is all about. Then we have cell division after that, after we get the two separate sets of DNA. So mitosis is when we copy the DNA and then divide the cell. The other part of the cell cycle is interphase. Now you probably, and I'm right there with you on this, when we first ever heard about mitosis at some point, we learned that the stages of mitosis were interphase, prophase, metaphase, anaphase, and telophase. Just sort of, you know, throw it out there, just sort of like rote memory, you know. Well, interphase is not part of mitosis. Interphase is when a cell's being a cell. Interphase is when a cell is busy doing cell things, whatever the cell's doing. If it's absorbing nutrients or if it's carrying red blood cells, if it's a muscle cell, if it's a nerve cell, whatever the cell's doing, that's during interphase. But interphase is not part of mitosis. Interphase represents about 99.9% of the cell cycle. Cells doing cell stuff. It's only when the cell moves in, replicates its DNA and gets ready to divide, does it go into mitosis. Now this is interphase. This is when a cell is busy being a cell. It's doing cell stuff. It might be generating energy from burning glucose. It might be making proteins. It might be producing enzymes to destroy bacteria or to break down food. It's maybe doing all these things. You know, this is interphase. Doesn't look like anything's happening, but it's probably pretty busy. You know, the nucleus is intact. The chromosomes are not visible because they have all unwound, but it's busy doing stuff, you know, whatever a cell's going to do. And it's going to spend a cell will spend about 99.9% of its time in interphase. Once we get out of interphase, we go into mitosis and cell division. Now, these are real-time videos of cells undergoing mitosis and cell division. You can see the chromosomes there separating, and they've copied themselves and they're moving apart to opposite ends of the cell. Here we can see cell division taking place where the cell's actually splitting up into two distinct daughter cells. And it's very, very rapid. The actual time spent in mitosis and cell division is usually measured in seconds, because you can see how rapidly it's occurring here. You know, most of our textbooks will show mitosis and cell division as taking up 1/4 of the whole cell cycle. Now, if a cell lives for 24 hours, it implies that the last 6 hours of its life is busy doing this, when it's actually about the last 6 seconds of its life before the daughter cells are formed. Now, the stages of mitosis, which is nuclear replication, and all living cells will go through mitosis if they have a nucleus and they're not stuck in a non-dividing state. As I said, the cells that don't undergo mitosis are the muscle cells and the nerve cells. They don't replicate. We're stuck with those. But bone cells undergo mitosis, they grow. Skin cells undergo mitosis. All the epithelial cells undergo mitosis. The connective tissue cells, undergo mitosis as long as, but the bone and the neurons don't. Everything else undergoes mitosis. So the stages of mitosis are always going to be the same. The idea is that we want to make perfect copies of the DNA. We want to copy the DNA and make it a perfect copy so there's no mistakes. We don't like mistakes in our cells. So the 4 stages of mitosis are prophase, metaphase, anaphase, and telophase. In prophase, the cell is getting ready to undergo mitosis and cell division. At this point, the chromosomes, the DNA condenses and the chromosomes become visible. There's little black squiggly lines there in the cell highlighted in red. Those are chromosomes. And what happened is that they have condensed. They become visible. It's a whole lot easier to move chromosomes around inside a cell if they're condensed into large structures. Otherwise, you're trying to move very tiny strands of large molecules, and it would be easy for them to get caught and twisted around and break apart. So we condense them into larger structures. The nucleus disappears. The nucleus breaks down and disappears because the chromosomes are forming, and the chromosomes have to move. They have to leave the area of the nucleus. We have duplicate sets of chromosomes at this point. We have copied the DNA. We've taken the original DNA and used that to build a perfect copy of the original DNA. We'll talk about how we did that when we get to the lecture. We end up with 2 perfect sets of the DNA. And if they weren't perfect, we're going to fix those problems. We have proteins that slide across the surface of our DNA looking for mistakes. Our error rate is somewhere in the neighborhood of 1 in a million on the DNA strand. But after these enzymes slide across the DNA structures itself, the error rate drops to something like 1 in a billion, which is really good, so that we don't copy mistakes. As we start copying mistakes, we're going to continue to pass that mistake on. We call it a mutation. We don't like mutations. So, okay, metaphase. The second stage is metaphase. Now we have the chromosomes. The chromosomes are very visible. They have migrated to the middle of the cell. They line up on the middle of the cell, so we guarantee that we have the right number of chromosomes. We're supposed to have 23 pairs of chromosomes. At this point in our cells, we would have 2 sets of 23 pairs. They line up because we want to divide them evenly. And it's a whole lot easier if they're lined up before, so we can pull them apart. So the chromosomes are in the middle of the cell, and they're ready to be pulled apart to opposite sides. This is the shortest phase. This is anaphase. In anaphase, the chromosomes split apart and start being pushed and pulled to opposite ends of the cells. You've heard about spindle fibers at some point, I'm sure. The little line there you see between the chromosomes. Spindle fibers form on both ends of the cell. They're pushing, some of them are pushing the cytoplasm apart. Some of them are pulling chromosomes. Some of them are pushing chromosomes. But what we end up with is cytoplasm being pushed apart and the chromosomes being pushed or pulled opposite ends until we end up with 23 pairs on the left and 23 pairs on the right, and they're identical. If they're not identical, everything stops. If we don't take the right number of chromosomes from one side to the other, everything stops. The cells will shut down if there's a mistake. Yeah. One of the things about our bodies is that there is a great deal of redundancy. We don't depend on just one location on DNA for our whole body to do something like blood type or hair color or eye color. We have hundreds and hundreds of locations. So a mistake in one area isn't going to hurt. We may just cut it out. But if it's a significant error, then the whole cell could stop right there. And then if we can't fix it, then we'll destroy the cell. So the chromosomes are evenly distributed. In a normal healthy cell, we would get the chromosomes evenly distributed to both sides, 23 pairs. Whatever the parent cell was, the two daughter cells are going to be identical. And then the last phase is telophase. In telophase, we start regenerating the nucleus. The chromosomes unwind. And then we start to we finish cytokinesis. Cytokinesis actually started in anaphase. Anaphase is the shortest of the phases. It's when they pull apart, and then we start dividing the cell. And you can see here, this is early telophase. Here we can start seeing the indentation of the line there. Here we have the daughter cells almost complete. What happens in cytokinesis is a band of protein forms around the outside of the very large cell. At this point, you're bringing the chromosomes to opposite ends. This band forms, and it's what we call a contractile protein. It's the same protein we have in our skeletal muscles, and it starts to contract and squeezes the cytoplasm apart. And then the two daughter cells become two distinct cells. And we essentially, it looks sort of like one of those balloon animals, you know, the kind that you can twist into different shapes. Only imagine a balloon animal, you twist it, then you can pull it apart. That's what happens here. The cleavage furrow formed by the protein that's squeezing just causes it to separate. And we have two brand new daughter cells with however long their lifespan is going to be before they have to go through this again. So, and that is the whole process of cytokinesis and mitosis. So cells are doing this all the time. You know, the average skin cell, like I said, lives for a couple of days. Red blood cells don't undergo mitosis, and they'll live for 4 months. So white blood cells, some will last for years. Most will last just for a few days. The ones that are responsible for our immunity, that develop what we call antibodies, will generally keep it. Some of those around for a lifetime. Others will be generated right away. Some only last for a few hours. Some will last for a few days, for a few weeks, and some will last a very long time. So it just depends. Questions on any of this? We'll spend a lot more time in this on a lecture. Now, let's go ahead and do the cheek cells. Okay, now, how are you going to do a cheek cell? Well, we spared absolutely no expense on this. We purchased Walmart toothpicks. Actually, it says diamond toothpicks. And what you're going to do is a very simple procedure. Oh, great! I wore a white shirt today too. I didn't even think about it. If you get this stuff on your skin, it'll wear off. You get it on your clothes. It's a permanent reminder of lab. I'll just tell you that. So, and of course, I wore a white shirt. Okay. The rubber slips. Now, the way we're going to do this is you're going to take a microscope slide. And the microscope slide has a frosted surface on there where it says specimen. And it doesn't matter if you have that up or down, but it works easier if it says specimen and you can read it. Otherwise, you're reading it backwards. But take the flat end of the toothpick, stick it in your mouth, scrape the side of your cheek, smear that on the glass of the cover slip, throw the toothpick away, put a drop of water on this. This is normal saline. And then put a drop of the dye on it. And now I've got a drop, a bubble there effect. Now take a cover slip, which is a little piece of plastic. Another in there, so I can feel it. Here it is. It's a little square of plastic, and take it and drop it gently on this bubble. And now I got a little, let me put the camera on this. Stop the sharing for a second. And this will give you a better picture of this. Let me bring it up to a larger size. Come on. Oh, whatever. See, there's the cover slip. And you can see it's laying on top of the water and the dye. And now if you put this under the microscope, you will be able to see cells. So trust me, it always works. So don't use the pointy end of the toothpick, it'll hurt. So anyway, we have microscopes all the way across the back of the room. I'd like everybody to get a slide and a toothpick and a cover slip and scrape away. And throw the toothpicks in the trash. Let me bring up the what you're looking for is ultimately something that looks like this when you get your cells. Now the microscopes are turned off, so you'll have to turn them on. They're all plugged in, they're all on low power. Just place it on the stage. If you, I'll walk around and see if you have troubles. Go ahead and take your slide. I'm sorry, I have your wrist. You don't take your slide yet. So. Yeah, it's up in the front there already. Just walk around behind my desk. There's two bottles there. Yeah. One drop of each and drop the cover slip on it. The cover slips are, did you get your cover slip?
Yeah.
Okay. Sounds good. There you go. Very good.
Sorry, I got up a bit late.
Well, that's okay. We're making doing the cheek cells now. So
Okay, cool.
Everything you need is over here. And just take the toothpick, scrape the inside of your cheek, smear it on the microscope slide, add a drop of dye to it with a little salt water
Okay.
and drop the cover slip on, and microscopes are available.
Wait, I think I forgot my cover slip maybe.
Let me see if we should have some more.
Wait, is it this? No, it's a
It's these little clear things here. Here we go. Yeah, I thought. There you go. Put these down here. Going there. The outside valves, of course, focus levels, the inside valves is fine.
Just drop it on there, it'll spray it. Okay.
Well, that's not too- that happens.
So it's a hundred stuff.
Yeah. Okay. Okay. Well, I didn't get that much. Oh, okay, good. Anybody have problems? Okay. Yes. Okay.
He says it's out of the way. He said it's out of the way. Because I've been drinking a lot of water.
Yeah. Okay. That's even better. Okay. Okay, so I guess everybody got to see their nucleus and their cells. This is the first time we've done this in 3 years. So anyway, this is a very simple test, a very simple little experiment to demonstrate that there is, you can see the nucleus, you can see the cytoplasm, you can see the edge of, you can see the edge of the cell. Can't see the cell membrane because of its microscopic structures, but you can see a lot by going through this. So, questions on this? Okay. Now, let's take a little time and because this wraps up everything we have to cover on Unit 1. And it's not gonna, if I go ahead and start talking about the bones of the skull, it won't take us that long. It'll give us, we'll be sort of on track then. All I gotta do is find out where. Here we go. Share screen. Okay, now. There we go. Okay. Okay, the human skull is part of our skeletal system. There are 206 bones in the human body, and there are 22 of them in our skulls. And it is not that challenging to learn these to learn the names of these bones. Now, I know our bone test is listed, whatever the date is, I'm going to probably slide that back a week too. The bone test is requires a lot of memorization. It will not be multiple choice. You'll have to know these structures. And so it's the kind of thing you learn a little bit every day on. You don't have to learn, can't learn the whole thing all at once. You got to learn a few bones every night. Pretty soon you'll discover that you have them all committed to memory. I say this from 19 years of experience of giving the bone test. And I always tell people, don't wait to the last minute. Always spend several weeks getting ready for this test because there's a lot to remember. The advantages of this is no matter what you're going into in any type of healthcare field, you're going to need to know bones. No matter what you're doing, you're going to need to know the bones. It's once you learn them, it's very hard to forget it, no matter how hard you try. You will remember, trust me, you will remember these bones. And the more you are, depending on what you're doing, the more daily application you have with them, the more you're going to remember them. So, but this is this is going to be your first exposure, I think, for many of you to these 206 bones. And of course, all the processes, openings, cavities, and bulges and bumps that stick up. So just to throw that out at you. Okay, this is what our skull looks like. This is one of those 360-degree x-rays like you get when you go to the dentist, it shows you all the bones in the skull. In this case, it also shows the bones of the vertebrae, and we can see the sternum and the top of the ribs too, but it does show you all of the bones of the skull and the cervical vertebrae. This is what X-ray is good for. X-ray is great on stuff that's not soft tissue. Okay. So the number one bone that nobody ever gets wrong on the bone test is the frontal bone. The frontal bone sits right here in the front. It is what we can feel if you rub your forehead. You can feel your frontal bone right there underneath that. The skin is very, very thin there. And this large bone starts about mid-level at the eye sockets and goes all the way back to about halfway back and comes down to right about the level of the top of the ears. So we have this very large bone covering the front of our skull. It gives us the shape of our face. It gives us the gives us the shape of the eyeballs. The muscles on top of this bone are engaged in expression. We don't use these muscles up here for much because they're very, very thin, but these are the expressive muscles that allow us to show, you know, whether we're happy, we're sad, we're annoyed, we're bored, things like that. The facial expressions take place along in here, but it is a very large bone, a very important bone. All the bones of the skull are designed to protect the brain. There's our there's our frontal bone right there. Now this next bone that we see is located in the eye orbit. The orbit is the opening that the eyeballs go into. Our eyeballs are about the size and shape of a ping pong ball, more or less, and they fit inside this socket or the orbit. The orbit is a very distinctive structure there. The shape of the orbit is often genetically determined by, sex, by genetic makeup. You know, different people have different shaped orbits. So now, on top of the orbit, there is a notch. There is a little groove up there. It's called the supraorbital notch. Now, supra is one of those terms that means above. So whenever we see the word supra or superior, it means above. So in this case, supraorbital tells you that it is a notch above the orbit. Supra, above. Remember those anatomical terms I mentioned in chapter 1, how you're going to get exposed to them as we go along. And this is the go along now. So supraorbital notch. Now I'll tell you, our bodies are very consistent. If we have the word supra, then we're going to have its counterpart, infra or inferior. So if we have a notch above the eye, we got to have something below the eye, and we do. And it's called the infraorbital foramen. The orbital tells you it's around the eye. The notch above the eye is supraorbital. Infra means below. Now the term foramen is one of those terms you're going to hear a lot. Foramen is an opening in bone, usually for a blood vessel or nerve to pass through. We have lots of foramens in our bodies, in our bones. The infraorbital foramen is a passageway for blood vessels and or nerves that usually probably goes out to the surface of the skin or directly underneath the surface of the skin. That is the infraorbital foramen. So whenever you have a superior, you're very likely going to have an inferior. If you, so we'll always look for that. Always look for this. The inferior is going to be a superior. Now here we have another, that arrow is not lined up, but very well. But that opening in the back there is the optic foramen. Anybody want to guess what goes through the optic foramen? The optic nerve, exactly. These foramens are designed to be passageways for either nerves or blood vessels or both. In this case, the optic foramen is designed to carry the optic nerve from the back of the eyeball into the brain. Now here we see a bone called the ethmoid bone. It's highlighted in green on that little animation that's going around. The ethmoid bone is on the inside wall of the eye socket. You could say it's on the medial surface, if it's near the midline, the medial surface of the orbit. And the ethmoid bone is more than just that little itty bitty bone you see highlighted in green there. The ethmoid bone goes all the way up into the base of the skull and all the way down. It actually divides our nasal cavity in half. It's part of that, you know, you'll see part of it there where we have that bone projecting there in the middle of the nasal cavity. I mean, there, you'll see it's a very large bone. Right now, it just looks like a little tiny bone because it's on the side of the medial surface of the socket. Of course, there's our frontal bone. You know, and yes, I put that on the bone test. Nobody ever gets it wrong. So that's one of your gimmies. So besides that, there's only so many ways you can ask, what's this bone in the front? Yeah. So, okay. On the side, we have what's known as the parietal bone. It's directly behind the frontal. It's on the either side on the top surface of the skull. And then directly below that, I don't know if it'll show up in the screen or not, we have what are known as the temporal bones. The temporal bones are where our ears are located. And it may or may not show you this next. Now this other one, this next bone here, the sphenoid bone, doesn't look like much from the outside, but it's very big on the inside. The sphenoid bone goes all the way from side to side of the skull. There's a sphenoid bone here. So there's my, yeah, here and here. It goes all the way through. It's right behind the eye sockets, right behind the orbits. So this bone, it's present on the exterior surface, but it will run and form part of the base of the skull on the inside. The pituitary gland, the master gland of the body, actually sits inside a depression in the sphenoid bone as it runs across the inside of the skull. And here we have the temporal bone on the side. So we have frontal in the front, two parietals, two temporals. They come down here in the sides, you know, the location of the ear or the two temporal bones. And this bone here, we can all feel, this is our cheekbone. Right here, you can feel it right below your eyes. This is the zygomatic bone. The zygomatic bone gives the shape to the bottom shape to your eye orbit. And it will extend back all the way along the side here. So it not only forms the base of the orbit, but it will extend back. And as I said, it forms your cheekbones, essentially. It can be, depending on your position of the cheekbone, is all genetic. Now, below this, we have two large bones that make up what we call the jaw bones. The upper jaw is the maxilla. The maxilla is attached to the skull. The lower jaw is the mandible. There's the mandible. Come on. The mandible is a separate bone. The mandible is the movable portion of our jaw. The maxilla, the upper jaw, doesn't move at all. It has teeth in it. We have half of our teeth are in the maxilla. The other half of the teeth are in our mandible. Our mandible is elevated, meaning it's brought up against the teeth in our upper jaw so that we can chew. And so the mandible is this large lower jaw here. The mandible has a junction in the middle called the mandibular symphysis. Now, a symphysis is one of those terms that means where bones come together. When we were in utero and our skeleton was made out of cartilage, we had two separate mandibles made out of cartilage. They grew together and then they got calcified and they formed one bone. Our frontal bone did the same thing. Our frontal bone had two halves, both made out of cartilage. They grew together and formed one large bone too. So our frontal bone started out initially as two segments of cartilage, and then as we developed, it grew together, calcified, and we have one large bone. Same thing applied right here where it grew together. We have other structures known as symphyses in our bodies where they didn't grow together. In the pelvic girdle, there is a structure in the front of the pelvic girdle where the two pubic bones come together and are separated by what's known as the pubic symphysis. There, that structure is made out of cartilage and it's designed to be a shock absorber. But here we don't care. The mandible didn't need a shock absorber. It doesn't need to be flexible. It's designed to chew. It's not designed to absorb shock. So here we see our the mandible, the mandibular symphysis, and the angle as it goes up. On the inside of the mandible, we have two openings. We have the mandibular foramens, one on each side. You can see the holes there. There's one on the other side. There's a close-up of this one down here. And again, the mandibular foramens are designed for passages of blood vessels and nerves. Now, there are some very prominent holes, foramens, in the mandible in the front. Those are not the mandibular foramen. It's an easy mistake. They're openings, but they're actually called the mental foramen. You'll see it here in a second. Okay, now, mandibular foramens right there. Now, where the bones come together in the skull, we have these little squiggly lines. We call them sutures. It's a type of joint. They actually at one point were very flexible. When we were in utero, there was all hyaline cartilage growing there, and our skull was sort of had a lot of give to it. In fact, we even had a soft spot at the top of our skull, and that made it very advantageous during childbirth, so that our skull had a little give, because we're trying to push the baby's head through an opening that's about the size, it's about the size of a quarter, and we're trying to get something the size of a baseball pushed through that opening, and then with the body of the baby coming out behind it, because babies come out head first, usually. So the suture lines didn't form completely solid, probably till well into our teens, and maybe even as late as our early 20s, but this is where the bones grow together. This is a type of joint, just like our shoulder joint or our fingers and toes and stuff like that, except this one doesn't move anymore. It used to be flexible, but it isn't now. We call it the coronal suture, very much like a coronal plane or a coronal cut. The coronal suture goes across the top of the skull, giving you an anterior and a posterior region. Oops, what? I stopped that. Okay, now. There's our frontal bone. There is our parietal bone. There's our temporal bone on the side there. There is the zygomatic bone coming back from below the orbits. Now we have a new bone. Bone at the base of the skull on the back. It goes from the back here, and you can feel a little notch back here, a bump, and that will go all the way around underneath the skull to about probably in a straight line with the coronal suture. That's the occipital bone. The occipital bone protects the occipital lobe. Temporal bone protects the temporal lobe. Parietal bone protects the parietal lobes, and the frontal bone protects the frontal lobes. You know, who got named first, doesn't matter. The occipital lobe is where our vision center is located, back here at the base of the brain. And we have a very strong occipital bone to protect that area for us. There's another suture. This is called the squamous suture that separates the parietal bone and the temporal bone. And we have another suture line here called the lambdoid suture, separating the occipital and the parietal. There's an easy bone to remember. It's our nasal bone. It's right here. It's the first part of our nose. Directly below that, we still have hyaline cartilage. Okay. Now this little process here is called the mastoid process. You can feel these. You put your fingers behind your ears. You can feel two bumps, one on each side, sticking out. That's the mastoid process. The mastoid process is what sets on top of, well, no, I'm sorry. The mastoid process is right next to the bumps that hold the skull onto the spinal column. The mastoid process is a point of attachment of muscles that turn our head to the left or to the right. That's part of what it does. The other thing the mastoid process does for us is it's an alternate route to our inner ear. The mastoid process is made out of spongy bone, and the spongy bone transmits sound waves very nicely. And we can we detect sound through the mastoid process. Not only do we get it, you know, if there's nothing wrong with our hearing, not only do we hear sounds through our ear, through the ear canal, through the tympanic membrane and the bones of the middle ear into the inner ear, and generate nerve impulses, but we can also get it through the mastoid process. And even just placing my fingers over the mastoid process, I think I sound different because I'm hearing myself differently by just doing this. Now, this is also the location of cochlear implants. A person, you know, a cochlear implant, and essentially an artificial external ear, goes in here, and then the signals are sent to the inner ear. And so often you'll see an individual with an implant in this bone back in here. So it has two distinct roles for us. One, it picks up sound, and two, it's a point of attachment from some very large muscles that allow us to turn our head to the left or to the right, or to nod our head up and down like this. So we can all feel them very easily. Okay. Now, in front of the mastoid process is a structure called the styloid process. The styloid process is a little pointy bone. That's its technical term, pointy-ended bone. That's what a stylus is. A stylus, the term stylus originally meant pencil or writing implement. Thousands of years ago, thousands of years ago, you can tell it's the first day back from the holiday, when early writing skills were developed, they would use tablets of clay, and they would use sharp pointed objects to write into the soft clay, and then it would harden, or they would bake it or whatever. So the term stylus meant a sharp object. Turntables have a needle called a stylus that you place on top of a record, for example. The styloid process is any structure on a bone that has a sharp pointed end. And we'll see two of them in the skull. We'll see them in both bones of the lower arm, are going to have styloid processes. What they end up doing, they become attachments for muscles. You know, they're very thin, very narrow, but they give muscles a place to attach. In terms of styloid, because they're sharp and pointy and narrow, they were named by early anatomists because they look just like a stylus. So, okay. This bone is the ethmoid bone, looking at it from the side, looking right into the eye socket. The ethmoid bone will go all the way across to the other side and will go up into the base of the brain, and it will go down into the base of the nasal cavity. It will form the back half of the nasal cavity. Directly in front of that is a bone called the lacrimal bone. You may have heard of lacrimal glands, where we have the tear ducts. Well, there's an opening in the lacrimal bone for the lacrimal gland to pass through. So because the lacrimal gland is designed to constantly wash our eyeballs again and again and again. So it sits directly in front of the ethmoid bone. Now there is that mental foramen, that little opening that I was telling you about in the mandible. For some reason, the front of the mandible is called the mental area. And so the foramen in the front is called the mental foramen. Wouldn't make sense to call it the mandibular foramen just because it's in the mandible now, would it? So that was sarcasm. So yeah, I have no idea why they, whoever won that argument really liked the mental foramen. I'll just tell you that. But that's the mental foramen, one on each side here. Again, passageway of blood vessels and nerves, because we have to keep our skin that covers the mandible alive. Okay, now there's another close-up of the styloid process in front of the mastoid process. This also shows you the temporal bones. And in the temporal bones, we have the openings that lead into the inner ear, the middle ear and the inner ear. There are some very pronounced structures here that lead in here. If you were to look at the skull, you could, this one's not a very good model, but you could see that the location of the opening for the ear canal going into the skull is directly behind the end of the zygomatic bone. And you can see it here in this illustration. There's the opening for the ear canal right here. This is the external opening of the ear canal going into the skull. If you have an external, got to have an internal. And we'll see inside the skull where the internal opening is located. So, there's your temporal bone, showing it in its full location. One thing I like about this is that as you go around, you can see the styloid process on both sides. And these openings directly above the styloid process are the external openings for the ear canal. And if you look carefully as it comes around, you'll see an opening there and there at the ends of the, you'll see like a ridge. And at the end of the ridge, in the middle of the skull, you'll see an opening. And that's the internal opening for the ear canal for the passage of the auditory nerve to go into the brain. Okay. Again, temporal bone from the outside. Now, the occipital bone back here. It gives us the shape we have at the back of our skull. It protects the occipital lobe, and there is a large opening in the occipital bone that gives us access to the base of the brainstem, a structure known as the Medulla Oblongata. The spinal cord comes up and fuses with the brainstem just below the Medulla Oblongata. The Medulla Oblongata is a major reflex center for our bodies. It controls our breathing rate. It can speed up our heart rate. It can slow down our heart rate. It doesn't control the heart, but it does have certain, it has the ability to at least tell it, hey, we should speed up the pace now. And there's a very large foramen in the occipital bone called the Foramen Magnum, where the spinal cord comes up to join the brainstem. Foramen Magnum means big hole. There's our occipital bone, and as it goes around, you can just see at the base of that, the Foramen Magnum there, just projecting through. Okay. I mentioned this already on sutures. So there are other sutures besides the lambdoid and coronal. We have the big one here in the front, in the middle, called the sagittal suture. So there's our frontal bone, parietal bone, occipital bone. There's the coronal suture, and here we have our sagittal suture. There's the lambdoid suture again. Now, I've mentioned the sphenoid bone and the ethmoid bone already. This is what they really look like inside the skull. The sphenoid bone extends from side to side. It extends up into this region in here. It spreads out across like this. It helps to form the base of the skull. The sphenoid bone is often described as the keystone bone of the cranium, or the batwing bone, or the Batman bone, or just the backbone, because of its very distinctive shape, because it doesn't take a lot of imagination to look at it and say, hey, it looks like a bat. This bone that sticks up here with this little process going up, it's something else about bones. If there's a bump or a protrusion or something, it's nearly always called a process. So, for want of a better term, this is part of the ethmoid bone over here. Now, there's our sphenoid bone. You can see it's a very large bone. It extends from side to side and down across, and forms part of the base of the skull too. It has these large wing-like structures that protrude in the sides, and that sort of gives it sort of its bat wing shape. Again, you can see the sphenoid bone has both a greater wing and a lesser, what we call a greater wing and a lesser wing. The greater wing and lesser wing, there's the lesser wing right there, and the greater wing is right below it. It's often been said that it's the lesser wing of the sphenoid that looks like a bat, but it's a very distinctive shape. This structure right here, oh, there's the Foramen Magnum. Okay, that structure right there in the middle, that is called the sella turcica. It's highlighted in red over on the side here. And the sella turcica is where we place our pituitary gland. Pituitary gland is the master gland of our body. It controls 8 different hormones. Hormones are chemical messengers that tell our bodies to do things. Like growth hormone, for example, is a hormone. It's a chemical messenger telling us to grow. Thyroid hormone is another chemical messenger that controls our metabolic rate. Pituitary gland controls both of those, as well as 6 others. And it is located in the sella turcica, probably the most protected portion of any structure inside the brain. Because the sella turcica means Turk's saddle. And because of its shape, it's shaped like a saddle. That's where the early anatomists gave the name to it. That is the sella turcica. The sella turcica is in the middle of the greater wing of the sphenoid bone. So now we can see there's the Foramen Magnum. There's the sella turcica. Also within the greater wing of the sphenoid, we have 4 openings, 4 foramens for the passage of nerves. And fortunately, they're the same on the left side and the right side. We have the foramen. Our first name is all foramen. We have the foramen ovalis on the outside and the left and right side. And we have the foramen lacerums in the middle. And the order is ovale, lacerum, lacerum ovale, or OLLO, Ollo. You can remember that. You can remember the order of these four foramens. They're all located in the sphenoid bone. Now, if we tilt the skull forward, we can expose some other openings here that we didn't expect to see. This angle, if we go back one slide here, we can see we're looking straight down. But if we tilt it, if we tilt the skull, if we were to tilt the skull forward at about a 45-degree angle with the tilt it forward at about a 45-degree angle, we're going to expose some other openings, because now we can see this opening here that we didn't see before. We're going to see this opening here, which we didn't see before. And we're going to make this opening here more prominent. So the first opening that we see is the optic foramen. That's what we saw early on. The optic foramen penetrates into the back of the eye socket, and the optic nerve is coming through that foramen. A straight-on shot doesn't give you that view very well. Here we see the foramen rotundum. This foramen disappears if you're looking straight down, but if you tilt it forward, you see it. Again, it's the passage of a nerve. These openings here are the internal acoustic meatus. They are the internal openings for the auditory nerve as it goes into the brain. The auditory nerve ran down along these two ridge lines from the inner ear where it formed and pops out here at the internal acoustic meatus to go into the brain. And this ridge line is called the petrous portion of the temporal bone. It is a ridge that you can see. And there's an opening there. The auditory nerve comes out into the base of the brain. Petrous portion of the temporal bone is located right there on those two ridges. Now this opening is the jugular foramen. There's one on each side. Our jugular veins drain the head and come down and return towards the heart. They pass through these two large openings on either side of the foramen magnum. Right there is the hypoglossal canal. The hypoglossal nerve passes through there. Now, hypoglossal nerve, a little opening for a very powerful nerve. That nerve controls our ability to swallow and speak. It innervates the tongue and innervates the ability to swallow. An individual who has had damage and it leads up to the medulla oblongata, the reflex center in the brainstem. A person who has had damage in the brainstem loses the ability to swallow. And they will often, the first indication that there's a problem there is that they will aspirate, they will choke on fluids or solid food that they're trying to eat. They won't be able to swallow it. They will end up gagging on it. Many of your older patients that are dealing with various types of dementias that can't communicate well, if at all, it's not uncommon for them to have a stroke, a damage in the medulla. And one of usually about the only indicator you get is that they're going to aspirate their food, or their solid or their liquid food, because they can't swallow. Because it's not one of those things where a person has damage to the medulla and you have any kind of overt sign. In many cases, the first overt sign is they develop pneumonia about 3 or 4 days after they started aspirating because they suck bacteria into their lungs. So it's hard to catch that. So the hypoglossal canal, very important passageway for the hypoglossal nerve, because that's what is controlling the movement of our tongue and our mouth, helping us to speak and to swallow. Okay. And there's that arrow should be further down. There's the jugular foramen again on this side. Right there. Oh, there we go. That's it. Oh, okay. There's the petrous portion. I jumped ahead of myself. That ridge line on either side is the petrous portion of the temporal bone. We have the internal opening for the auditory nerve coming out right there. There's our jugular foramen on each side, right there. Now, the ethmoid bone, the one that we saw on the medial side of the eye socket, also extends upward into the base of the skull. And up there, it's perforated. The ethmoid bone is perforated. It sits directly above the olfactory bulbs, which are helping us to detect odors. Now, in our nasal cavity, at the top of our nasal cavity on the inside structures, we have olfactory receptors, and our olfactory nerves pass up through this cribriform plate structure of the ethmoid bone. It is a perforated structure, and these little tiny branches of the olfactory nerve go up through those openings to the olfactory bulb. That's how we get signals from our nasal cavity that I smell something passing up into our brain. And so then we have to process it and say, oh, I know what that is. You know, I recognize, you know, how do we recognize, otherwise, how do we recognize odors? You know, baking bread, how do we know what baking bread is if we've never smelled it before? You know, or flowers, or different types of any kind of food, or chemical odors. You know, we have to store those odors in our memory. We find that memories and odors work very closely together to, you know, give us a better picture of things. Anyway, the top of the ethmoid bone is the cribriform plate. It's got perforations in it to allow these olfactory nerves to come up. And if you look very carefully, you can see some little tiny holes in that ethmoid bone. And above the ethmoid, there is a structure known as the crista galli. That's this thing sticking up here, this little pointy structure. The crista galli sticks up in the middle of the cribriform plate, and it's the point of attachment of the meninges that surround our brain. The meninges are connective tissue that allow the brain to float in cerebrospinal fluid. Our brain is not strong enough to support its own weight, and it would collapse. But what we do is we surround it by these 3 layers of connective tissue filled with cerebrospinal fluid. The brain floats, and we attach them, and they're called the meninges, and the meninges are attached to the crista galli, so our brain doesn't go sloshing on around on the inside of our skull. So what's it look like? Well, the animation here shows you the cribriform plate is in green with the olfactory nerves next to it. That's for our olfactory responses. This thing labeled 58 is the crista galli, right there, where the meninges attach. Crista galli, and then the cribriform plate. Cribriform plate is right there. And so we bring up the branches of the olfactory nerves, come up through the perforations in the cribriform plate, connect to the olfactory nerve there. It's convenient since the receptors are right here inside the nasal cavity. Now, on the underside of the skull, we see a few things. We see the carotid canal right next to the jugular foramen. That's where the carotid artery goes up into the brain, and the jugular vein comes down through the jugular foramen right next to it. This mandibular fossa. Fossa is a new word for us. Fossa means flat. Flat area. So we have a foramen as our opening. Fossa is our flat area. And that's where the end of the mandible articulates. Articulate means the bones come together. the end of the mandible articulates at the mandibular fossa. There's the vomer. Vomer is a bone that divides the nasal cavity in half. We like to have turbulence when we breathe. It allows the air in our nasal cavity to warm up and be exposed to mucus, to be cleaned up, and also to become moist. And so the vomer is part of this structure that divides the nasal cavity into two separate regions. Here we have the palatine bone directly in front of the vomer. The palatine bone gives rise to the roof of our mouth. The roof of our mouth is called is formed from what's known as the palatine process of the maxilla. There's the palatine process of the maxilla. It came off the palatine bone located right here. And that is what we would refer to as the roof of our mouth. We could feel that when we stick our finger in our mouth and push up. That's our hard palate. There, that's the palatine process of the maxilla. There's our vomer again, and the foramen magnum again, and our styloid process again, our mastoid process. Now these occipital condyles, another new word, condyles. Condyle is a bump that goes superior, inferior. In other words, it goes up and down. The condyle is a process on the bone that will go up and down. Its structure is straight up and down. Here we see the occipital condyles. This is where the skull sits on top of the spinal column. This is where the skull sits on cervical vertebrae number 1, the top of the spinal column, right here. This first bone. Right here, cervical vertebrae number 1, or C1. It's called the atlas, and it holds the skull up, and the occipital condyle is set on top of the atlas. And so those condyles rest on top, rest on cervical vertebrae number 1. Now, the reason I made such a big deal about a condyle having a bump that goes up and down is because next week we'll see something called the epicondyles. It's also a bump that goes lateral medial, meaning side to side. We'll have a medial epicondyle and a lateral epicondyle. It's just a bump. It just doesn't stick out from the side of the bone. Does it stick out towards the top of the bone? Okay. And the structure in the middle, right above the nasal bone, is called the glabella. That's all that's left of of this two separate structures. This represents where the frontal bones grew together, is at the glabella. And there's the other part of the ethmoid. This divides the nasal cavity in the front. The vomer divides it in the back. The rest of the ethmoid divides it in the front into two separate structures. So we want to have turbulence. We want to have that air moving around in there. Okay. Now there is the external opening of the auditory nerve. You know, from the inner ear, we go into the auditory nerve, and it runs through here, through that opening there, through the petrous portion of the temporal bone, and pops out on the internal auditory meatus or acoustic meatus, because if you have an external, you got to have an internal. Okay. On the fetal skull, we see a few structures here of interest. We see there's the frontal suture. that we don't see in the adult skull. The frontal bones grew together. We don't have that suture line. What we do see here is our what we call the soft spot, the anterior fontanelle. That would be where the frontal bones and the parietal bones that have their junction. That eventually calcifies and forms solid bone and becomes rigid, immovable. But we have our soft spot conveniently for us at childbirth so that we have a little flexibility in our skull when we're passing through the birth canal. Frontal bones, parietal bones. Here we see the parietal bone and the frontal bone, temporal bone. The bones are formed, but in the case of the frontal bones, are growing together. Now, when we are in utero, we have a series of fontanelles. The big one is the anterior fontanelle, but we're also going to have a posterior fontanelle and a sphenoidal fontanelle and a mastoid fontanelle. So we're going to have about 5 different fontanelles altogether. There's our coronal suture. There is our sphenoidal fontanelle. It closes too, but it gives us a little bit of flexibility from side to side. There's our mastoid fontanelle, flexibility in the back. There's the anterior fontanelle right on top. It's the big one. Frontal suture, which goes away. Coronal suture and sagittal suture, which don't go away. And we look at the back, the posterior surface of the skull, we can see the posterior fontanelle. Sagittal suture, parietal bones, lambdoid suture, and posterior fontanelle. Because if we have an anterior anything, we've got to have a posterior. So, and there we go. That's our skull. Now, next week, I will have the lab test is due next week. I've given it a week. If you wish to take it on ground, let me know. I'll have hard copies available. And after lab, you can arrange with Jennifer, the tutor, when you'd like to take it. She'll have the hard copies with her. So it's either way you want to do it, online or on ground is fine with me. I just don't want to take the class time for the test because we got a lot to cover. So, next week in lab, we're gonna take a closer look at the skull. I'm sure nobody was thinking hard about it today, and I understand that. I'll have the models out for you all, and we'll do the skull and we'll do the axial skeleton, which is the vertical axis, and we'll move right along. So, any questions? You look like you're going to ask me a question. You look like you were going to have a question.
No, I don't think so.
Okay, well, that's
Probably when
fine.
I walk out the door.
We're always open. Send me an email. So we never sleep. Okay, I think we're done then. Let's get out of here.
I'm sorry.
Well, let's go. Oh, I will. Here it is. I'll be glad to.
What did I miss the first?
Oh, I had talked about mitosis and cell division. It'll be on.
Okay.
It'll be on the recording.
Okay, awesome.
Okay.
And then I
Yeah.
always forget about the recording, but
That's okay.
I'll go back.
Sure. Go ahead and watch it. Then you only have to watch the first 10 minutes or so.
Okay.
Okay.
Thank you.
Mhm.